So I'm really glad that today we have Dr. Luca Fusi, which works at the Randall Division or Randall Center for Cell and Molecular Biophysics. And Luca does very interesting experiments trying to combine mechanical forces with hard mechanics, and it's really nice the way that he integrates all this different technology to answer a very sort of important and robust biological question. So this Luca's talk will complement all the sort of the different master classes that you had at the single molecule level that Rafa gave you, at the cellular level that Alberto gave you. So in a sense, it's a nice way to combine sort of it lies within those scales, so to speak, and even beyond at the larger scale. So it's a very nice sort of complement to with a different technique of what you have been seeing so far. So I'm sure you will love it. Again, thank you, Luca, for agreeing to give this class. We're very grateful. And the stage is yours. And yeah, good luck with it. Thank you very much, everyone.
So thanks again, Sergi, for inviting me to give this seminar. It's a great honor to be here. And today, the aim of my talk is to present some of our research on muscle contractility, but also with a focus on the techniques that we have developed in the lab to answer a specific question. And I think, as Sergio was saying, I think the techniques that you're going to see today are complementary to what Rafa has shown you in the previous talk about at the single molecule level. In fact, probably we will use the same terminology for defining some mechanical concepts. And the other thing I want to say is that if I would like to this lecture to be a bit interactive. So if you want to stop me anytime, please do it and ask your question. Otherwise, we'll do at the end. And so the subject of our study is muscle, and we want to understand how the muscle works at the molecular level. And in particular for mechanobiologists, muscle is very intriguing because it's not only a tissue that's been evolved to generate force and shortening, but it's also a tissue that is exposed to a wide range of forces in nature, and those forces can act as can be transduced by the muscle cell to trigger some pathway, trophic pathway, for example, to increase the muscle mass in response to exercise. So everybody knows that if you exercise, your muscle mass will increase, and vice versa. If you don't exercise, or if you are immobilized, your muscle will get wasted. And so there is a lot of interest in trying to understand how the forces can be transduced into biochemical signals. But that's not the focus of this talk. And this talk is more about on a short time scale, what is the effect of forces on the mechanical performance of muscle. So here you have an example of this, and you have an example of a simple exercise, which is jumping on a platform, and you see the force which is developed by the leg muscle in different phases of this exercise. And you can see that while the subject is standing, the leg muscle produces a force which is equal to the body weight. But then when the person wants to jump, the muscle generate larger force, and then during the flying phase, there is no force applied to the muscle at all, so the muscle is unloaded. And but what happens at the end of the jump, during the landing phase, you have a massive increase in force on the muscle of your legs, and so in this case, the muscle are forced to act not as force generator but as dampers. You have to dump a lot of energy, external energy, and so and you can see that the force absorbed by the muscle is about 4 times what they have in normal conditions. So the second message of this is that you have the muscle cells are exposed to large forces during concentric contractions, so when you have shortening, or during eccentric contraction when you have a stretch of your muscle. And these schematics here represent the hierarchical organization of the muscle tissue. So the muscle is an organ which is attached by the tendon to the bone, And the muscle is made of bundles of elongated cells called muscle fibers. These are giant multinucleated cells. And inside of each of these cells, there are bundles of filament organized to form myofibrils. And in each myofibril, you have periodic structure, which are called sarcomere, that are repeated thousands of times along the length of the myofibril. And here you have a representation of the sarcomere structure, which is the unitary element that generates force in muscle, in striated muscle. And the sarcomere is made of thick filaments anchored to the M line in the center of the sarcomere. And these filaments are overlapped by thin filaments, which are mainly containing actin and tropomyosin, troponin. And there are two antiparallel filaments anchored to the Z disc, the boundaries of the sarcomere. And each arrays of filament are pulled by the myosin filament towards the middle of the sarcomere. So that's how force and shortening is generated at the sarcomere level. So our question is related to how the mechanical load applied on the sarcomere can affect muscle performance at the molecular level. And when we talk about the molecules that generate contraction, the main player is myosin 2, because this is the molecular motor of skeletal and striated muscle. And the single myosin 2 molecule is made of a long coil tail, which is about 150 nanometer. And then you have these two globular regions, which are the motor domain of the myosin molecule. And each of these basically use the ATP hydrolysis in the energy liberated by the hydrolysis to generate force and shortening. So here on the right, you have a representation of the crystal structure of the globular domain of myosin, which was defined in '93 by in this paper. And here there are some important domains that we have to define. So here, this is the globular domain in gray is where we have the catalytic domains, where the ATP is hydrolyzed, and also where the actin binding site is. And then we have a long alpha helix, which is coming out of the catalytic domain, which is where two other peptides wrap at these two positions. And these two peptides are called essential light chain and regulatory light chain. And these two light chain plus the heavy chain, the long alpha helix of the heavy chain, form what is called the lever arm, because then the myosin, during the ATP hydrolysis, the energy is converted in a tilting motion of this lever arm, and this is called working stroke, and it's an elementary step of force generation. Now, going back to the myosin molecules, those molecules are now in the muscle. They are assembled to form bipolar filaments, which you have a representation here. And so you have a region where there are no myosin motor, which is called the pair zone. And then on the two sides of these filaments, you have two arrays, antipolar arrays of myosin motors, which pull the actin in opposite direction towards the middle of the sarcomere of the thick filament. And so moving on, then we can say that it's now clear that the regulation of muscle contraction is performed at the level of the two filaments. That's why we call it as a dual filament mechanism of regulation in striated muscle. And that has become clear only recently, only in the last decade. And in fact, if you open up any textbook about muscle contraction, you would see that the muscle regulation is principally based on the regulation of the thin filament. So the thin filament has some accessory protein called troponin tropomyosin. The tropomyosin is represented by these strands wrap around the thin around the actin filament, and then you have this complex, which is called the troponin complex, and is made of three subunits. The TNC, which bind to calcium, that's the calcium sensor, and then you have the TNI, which is the inhibitory region of troponin, which has this long extension, which push the tropomyosin basically and keep the tropomyosin in a blocking position. So in what is called the steric model of muscle regulation, the tropomyosin position is biased by troponin to mask the actin binding site for myosin on the actin filament. That's why the myosin cannot bind and cannot generate force. But when calcium is bound to troponin C, this triggers a series of structural changes that lead to the removal of this inhibition by the TNI segment, and now the tropomyosin can move azimutally, and therefore the myosin binding region are exposed, and the myosin motors can bind and generate force. So that's the textbook view, which has been clarified by electron microscopy only recently. But also electron microscopy study have also shown that the myosin motor has also another type of regulation, which is at the carried out at the level of the myosin motor, and that the first evidence comes from the fact that this reconstruction of the thick filament, you can see the myosin motor which are not available because they are basically shut down on the thick filament surface by interaction with the filament backbone. And the two myosin motor of each molecule form this asymmetric motif, which is called an interactive head motif, and that inhibits the myosin ATPase. So in the resting muscle, basically the myosin motor cannot hydrolyze ATP because they are locked into this conformation, and they cannot even interact with actin. And also because actin is blocked by in the absence of calcium by the tropomyosin strand. So it means now that there are two different mechanisms of regulation, one at the level of the thin filament, one at the level of the thick filament, and at the level of the myosin motor. And that's what we are trying to understand. But what we know is that the trigger for muscle contraction is given by calcium, by a transient increase in calcium concentration in the muscle cell, which is triggered by an action potential, which is delivered to the muscle cell by the net axon of a neuron. And this electrical stimulus is converted into a calcium release from intracellular stores, and this calcium binds to the thin filament, triggering the activation of the thin filament. And that's what people have measured inside the muscle cell using calcium indicator. So you can see that the calcium concentration in the cytoplasm increased very rapidly in a millisecond time scale, and this is followed by force generation. And that's produced by a single action potential in the muscle cell. So now the fact that we observe an off state for myosin, in which the myosin motor are packed along in helical tracks on the thick filament surface, and an off state for in the thin filament where tropomyosin strand block the actin binding site for myosin. Then we have a question about how the two filament gets switched on and how fast. And so in a fully activated muscle, then you have the thin filament completely activated by calcium binding to troponin, and the myosin motor also are released from the filament surface. But now the question are how the dynamics of the filament activation control the force, and how the myosin motors can switch on. What's the mechanism? And more importantly, how these structural changes in both filaments control the force generation in the skeletal muscle cell. So to answer these questions, I think we have developed a very unique approach to understand the mechanochemistry and the regulation of the myosin motor in situ. And so we basically our approach is based on a combination of structural technique, which is based on fluorescent, called that fluorescent polarization microscopy, and then a functional approach, which is based on mechanical measurement at the single muscle cell level. And then we couple with cage compound photolysis to investigate dynamics, so to reproduce the physiological calcium transient or near physiological calcium transient in a muscle cell by using a photolysis of a cage compound, which is called cage calcium. So basically it's a compound that binds, that chelates calcium, so basically trapped calcium in this cage, chemical cage, and the calcium then can be released and be active upon UV stimulation. And so the fluorescent polarization technique is is a very unusual way of using fluorescence, because you might have heard of many other techniques based on fluorescence. For example, you have used, you are aware of super resolution techniques that allow to detect the position of single molecules inside cells or in solution, and so that's an application of that we can call as I mean, that is for localization of fluorophores. But then you certainly know another application, which is FRET, and that's an application for measuring distances in the nanometer scale, and measure interaction between proteins which have been labeled with fluorophore. So in that way, the fluorescence acts as a molecular ruler to measure distances. But actually, we measure fluorescence in this approach to measure conformational changes in the myosin motor, and we'll see how. So I like to define this fluorescent polarization microscopy as a molecular goniometer. So FRET is a molecular ruler, fluorescent polarization microscopy is a molecular goniometer. And that's because it's a very powerful approach to measure angular changes in protein domains, and particularly useful for studying myosin, because as it might be clear now, is that there are two classes of structural changes in myosin that involve a change, an angular or a conformational change in the myosin motor of the myosin molecule. For example, I've shown you how the myosin motor can be regulated and can be activated from an off to the on state. So in the off state, the myosin motors are folded on the on the coil tails, which is embedded in the filament backbone. And to be activated, the myosin motor have to undergo a large swing, and then become more parallel conformation to the filament to more perpendicular orientation that allows them to interact with the overlapping actins. So this in a conformational change that we can measure with our goniometer. But then there are another class of structural changes in the myosin motor which are more associated with fourth generation. And here you have the structure of a segment of actin filament, which is bound to the myosin motor. And here the myosin motor is shown in two different conformations. So one conformation with the lever up, which means that the myosin motor is ready to perform its working stroke, so is at the beginning of the working stroke. And then in the second structure, the myosin motor has tilted the lever arm by 70 degrees, and that's the end point of the working stroke. So basically, the working stroke, or the unitary step of fourth generation, consists of an angular change of the lever arm domain by 70 degrees. So again, here the molecular goniometer is important to measure in situ, in the muscle fiber, the amplitude of this structural change at the level of the myosin motor. But here again, concept of fluorescent polarization of a molecular goniometer. And basically the essence of this approach is to use a particular class of fluorescent probes, which have been developed in our lab, and these are called bifunctional rhodamine. So the difference with a conventional rhodamine is that this molecule have two attachment points. So they have two functional groups that can react with cysteines that have been introduced specifically in the sequence of a protein that we can easily exchange in the muscle. And in particular, here in cyan, we represent the regulatory light chain of myosin, and with different probes. And the two spheres for each probe represent the attachment points for the molecule, for the rhodamine molecule, and the segment connecting these spheres is actually is marking the orientation of the fluorescent dipole of the molecule. So because the molecule has two attachment points for each probe, then it means that the fluorescent dipole has been constrained with a specific orientation in the molecular frame. And therefore, with fluorescent polarization, we can excite the fluorophore with two different polarization, parallel and perpendicular to the filament or fiber axis, and the absorption of the photons from excitation depends on the relative orientation of the polarization of the excitation and the relative orientation with the fluorescent dipole. And then the emission of the fluorescent emission is also show a high level of anisotropy, because the fluorophore has a specific orientation in the muscle fiber. Therefore, if you measure parallel emission and the perpendicular emission, you will get two different intensity depending on the orientation of the fluorophore. So by combining this measurement and by combining information from different probes applied on the muscle, on the different points of the regulatory light chain, we can calculate two angles which are called beta, which is the angle of the lever of the basically the lever arm axis of the myosin motor with the filament axis, and then gamma, which is a rotation rotation angle around this axis, which is going through the E-helix of the RLC. So we basically describe the orientation of the myosin in terms of two angular coordinates. And that's what we have done in situ in muscle fiber in this study, where we used a set of 4 probes labeling the C lobe of the RLC. And we use RLC because this peptide can be easily exchanged in the muscle. So there's basically a hard work in the lab to produce this and purify this labeled RLC, which has been expressed in E. coli. And then once they are purified, they can be exchanged in the muscle. So you can replace the native RLC and exchange it with an RLC which has been labeled with one of these fluorophore, and then one fluorophore at a time can be used for fluorescent polarization. And then once you collect the information for all the 4 different probes, you can measure the angle beta and gamma. And then you can build a probability map for each beta gamma orientation and plot them in these spherical plots. So in the spherical plot, basically the latitude represents the angle beta, which again is the angle of the lever arm with respect to the filament axis, and gamma is the rotation around these axes. And then you can see that if you take a fiber at low temperature, which is unphysiological, the myosin motor assume these are all in this conformation, which we call it RX because it's relax conformation 1, so in the absence of calcium. And this conformation has a beta about 90 degrees, so it sits on the equator, as you can see. 90 degree means that the motor is more or less as we show it here, so perpendicular to the filament axis. But as we increase the temperature towards the physiological value, which is the body temperature around 30-35 degrees, we get the appearance of a new population at the pole of the sphere, and we call this population RX and RX2 and 3. And those populations have a beta angle of about 180 or 150 degrees. So it means that this angle has increased. It means that the myosin motor had moved towards the parallel orientation to the filament axis. And if we take the cryo-M model for the interactive hemotility, so the folded say of the myosin motor, we can calculate what's the orientation for the RLC, the 2 RLC in this orientation. You can see that they closely match what we found in the probability map for the RLC orientation in a muscle fiber. So this confirmed that basically when we cool the muscle, we induce a tilting of the myosin motor from this perpendicular conformation to a more parallel conformation, which is consistent with the electron microscopy model. So in other words, in a physiological condition, in a muscle fiber, the myosin motor adopt this conformation. Most of the myosin motor are folded in this conformation. So now the question is, what are the signals that induce the activation of the myosin motor in a physiological contraction? And so in the next experiment, then I will introduce the second approach, which is sarcomere level mechanics in muscle fibers. So this approach allows us to basically isolate a single fiber from the muscle, mount the fiber in a muscle chamber, in an experimental chamber where we can control the physiological parameters, the temperature, and so on. And then we can, the fiber is connected basically to a motor, which allows to apply very fast mechanical perturbation to the muscle fiber. And on the other side, the muscle is connected to the force transducer, which is a very is not commercially available transducer and motor, because they have high resonant frequencies, which means that these tools have been developed in some labs to apply very fast and record very fast mechanical perturbation. And then we can use different methods to measure also the sarcomer length in a fiber during contraction or during different mechanical states. And we combine this approach with fluorescent polarization. So in addition to this, we now introduce our fluorophore, introduce our probes, RLC, labeled with our bifunctional rhodamine. We can excite the fluorophore and record in our microscope the fluorescent polarization, and therefore detect the angles of the fiber in the muscle cell during contraction. So the experiment that we did here was to test the hypothesis that the myosin motors can be activated from their folded conformation by a mechanical stimulus, and meaning that the force applied on the thick filament can switch the myosin motor on, independently of calcium. So to do this, then we took a muscle fiber in the relaxed state, so in the absence of calcium, and we applied a series of stretches to the fiber, which increased the sarcomer length from about 2 micron up and above 3.3, 3.4 micron. And this elongation, if you look at the sarcomer structure, caused the elongation of the titin molecule. So titin is the third filament of the muscle sarcomer. It's a molecule that connects the Z disc, so the boundary of the sarcomer to the tip of the thick filament, and then it runs along the thick filament and connects the other end to the M band, so to the middle of the sarcomer. So this is a huge molecule, single molecule that runs from the M band to the Z disc, and it acts as in relaxed muscle as a passive spring. So if you stretch the muscle, basically in this condition, you stretch the titin links, and this passive force is directly applied to the thick filament tip and to the rest of the filament backbone. So that's the force that you can record with the force transducer during this mechanical protocol, and you can see that there is an increase as you stretch, and then followed by what we call viscoelastic decrease, and that's a typical response of stretching the titin molecule. So in an elastic response followed by a viscoelastic relaxation. And then we, in our microscope, we measure at the same time the fluorescent the fluorescent polarization of the 4 different probes on the C lobe of the RLC, and we can see, especially in this probe here, which is the one that is parallel to the myosin liver arm, you can see that the probe changed the order parameter in response to the force applied on the filament by titin. And this parameter here is directly linked to the mean angle of the probe with respect to the filament axis. So when this parameter goes down, means that the probe is becoming more perpendicular. So this means that the probe is highly parallel, because the relaxed state, because the myosin motor are folded on filament surface. As you stretch, the probe becomes more perpendicular with this typical, with a response which is mirroring the response that we see in force. And so this triggered the idea that that the myosin motor are now sensitive to the passive force applied on the thick filament, and this is visualized in our spherical plots as a change in the redistribution of population of myosin motor from this parallel orientation in the absence of loads to an equal redistribution between perpendicular and parallel orientations. So it means that part of the folded myosin motor have been transferred to the perpendicular state simply by the force applied on the thick filament. So now, if we want to characterize better the dependence on force, then we have to use another approach, which probably have already discussed in another lecture, and that's called force clamp mode. And so our mechanical approach allows us to generate a force clamp at the muscle fiber level. And so in this force feedback, basically we have the muscle fiber, which is attached to the lever of a length transducer, which is our motor. And then we have a force transducer to measure the force. And if you have a fast length transducer and a fast force transducer, you're able to generate very fast force feedback, meaning that you can control, rather than the length, you can control the force generated by the fiber. So here you have an example in an intact fiber, which has electrical stimulated to generate an isometric force, which means that the fiber contracts to the maximum force allowed for that length of the sarcomer. And then suddenly you enter the force clamp mode, and the system basically hold that force until there is a command, external command, which is applied to increase the force above the isometric load. And so we can choose the amplitude of that force step. That could be, for example, 15, 25, 40% or higher than the isometric force. But what is interesting is that you can do that very fast. You can, in a 0.1 millisecond, you can increase the force applied on the fiber. And so you can detect multiple phases in the response to the fiber to this sudden increase in load. And you can study how the sarcomer change and the protein in the muscle change their conformation at constant load. And then here you have the elongation of the sarcomer that is needed and is applied by the motor in order to keep the force constant. So we basically use this approach here to study what's the effect of tightening force on the thick filament structure in a muscle cell. And we did that in a relaxed fiber because we wanted to keep the thin filament off and just see what happened at the myosin motor level using our probes when we applied such a force pulse to the thick filament via tightening. And that's the force clamp mode applied on the relaxed fiber. So we can apply different force pulses, and we can keep the force high for about 50 milliseconds. The fourth step is complete about 0.2 millisecond, and this is the corresponding elongation which we have to apply to the sarcomer in order to keep that force constant. And so you have a very fast elastic elongation at the beginning, force followed by like an exponential stretch, which should counteract, if you remember, that force decay that we saw before. And that's how the system is able to keep the force, the tightening force, constant applied on the thick filament backbone. And then we look at our angular parameter, which is called the P2 for one of the probe in the RLC lobe. And then we see that the probe becomes more perpendicular, not in the phase one, so not in the very fast response, the 0.1 millisecond response to the step, but in the following phase, so in what we call the phase two. So phase one is the elastic response, phase two is the viscoelastic phase, where you see this exponential elongation. And what we see is that the orientation of the motor become more perpendicular as we applied a larger load. So the amplitude of the structural change is a plot here for phase two and phase one. So you can see phase one, there is no change, but in phase two, there is a change which is linearly related to the force, to the tightening base force applied on the thick filament backbone in the force clamp condition. And the other information that we can extract from this experiment is about the rate of this process, the rate of the structural change. And this is shown here by measuring the decay of these curves at each different load imposed on the muscle fiber. And we can see that at low load, this is about 100 per second, and the structural change become much faster when you apply a larger load on the filament. So there is a load dependence not only for the amplitude of the structural change, but also for the rate of the structural change. So this load dependence of the structural change in myosin allows us to define the idea that the myosin motors on the thick filament are activated simply by the stress applied on the thick filament independently of calcium. And that's a mechanism we call mechanosensing. So the thick filament myosin filament in skeletal muscle acts as a force sensor and can quickly react to changes in load by activating the myosin motor from the fold state depending on the load. And that from the fluorescent measurement we can extract a parameter of or which is linked to the level of activation, to the degree of myosin motor activation. And while in a resting muscle, so when you have zero force applying the thick filament, most of the myosin motors are folded, then you can induce activation simply by applying passive force, so by elongating the muscle, by stretching the sarcomere and stretching the tightening links. And then this trigger activation of the myosin motor. But you can also add calcium and keep the length constant, and then the force generated by the myosin motor itself at each level of calcium generate a graded release of myosin motor from the folded state, depending on how much force is applied on the thick filament backbone. And that's what we call the mechanosensing idea. So interestingly, we wanted to characterize recently this response in cardiac muscle too, because the cardiac muscle is another example of striated muscle. And so the players in the cardiac sarcomere are the same proteins, but we have different isoforms. So we want to understand what's the function of the different isoform and how they tune the different performance of the muscle to the physiological task. And so we did similar measurement by stretching the sarcomere in cardiac muscle. In cardiac muscle, the titin molecule is much shorter, and therefore this constrains the range of sarcomere length that we can explore in cardiac muscle. So it's, instead of going from 2 to 3 micron or more in skeletal, we can only stretch the sarcomere by only 300 nanometer, so from 2 to about 2.3. And the titin-based passive tension in that range of sarcomere length is not that much. It's about maximum 50 piconewton per thick filament. And in that force range, the titin molecule, therefore the force generated by the titin molecule is not enough to trigger an activation of the myosin motor. But when we add calcium to the system, so in the presence of calcium, then we observe different phenomena. So we observe that the myosin motor gets activated with a very steep dependence on the force, of the active force applied on the muscle. And that's what we define in this paper as a cardiac mechanosensing, which has a steepness different from what we found in skeletal muscle. And we think that this has to do with the force range, different force range which is applied on the different thick filaments in skeletal and cardiac muscle. And in particular in the heart, the force range is smaller, and therefore we think that the different isoform can tune the mechanosensing transition for the active force applied during the physiological heartbeat. Now, I think we come to the final part of this seminar, and in this final part, I will tell you about the last technique that we developed, and that's about cage calcium photolysis combined with the mechanics of single muscle cell and structural technique, the fluorescent polarization approach, to perform time-resolved studies of myofilament structure in skeletal muscle cell. And so here you have a diagram showing the myosin motor and the troponin complex on thick and thin filament respectively in relaxed muscle. So in relaxed muscle means that the calcium is not available, and in our assay, basically the calcium is trapped by the cage calcium. And so in this case, the myosin motors are folded back on the filament surface. We can stick a probe, bifunctional probe, on the regulatory light chain, and this orientation is almost parallel to the filament, as I've shown you in the previous slide. And then what we can do is also to place probes on the troponin C, which is the calcium sensor of troponin complex, and we can do it in two different positions, which is the N lobe, so the regulatory lobe of troponin C, and the C lobe, which is the embedded in the troponin complex. So why we chose this point? So first of all, because the TNC is the calcium sensor, and it has the probe here is very close to the calcium binding site on troponin. So this probe is expected to give us some information about the velocity of the structural change triggered by calcium binding to troponin. Whereas the other probe we placed in this position because this C lobe is part of the of a scaffold, of the troponin scaffold, which is then connected by the TNT to the tropomyosin strand. So this is actually expected to give us some information about the downstream events following calcium binding to troponin C. So now, with these three probes, this set of probes on RLC and troponin C, we are able to dissect the dynamics of the structural changes triggered by calcium in a muscle cell. And we can do that because with the photolysis of the cage calcium generated by UV laser pulse, we can quickly increase the calcium concentration in the muscle cell, and we can then study the dynamics of the structural change in troponin and those compare with those in the regulatory light chain of myosin. And then we can do that very fast because the fluorescence allows us to measure with high times resolution, 0.1 millisecond, the change in the polarization of the probes. So here we have an example of it. So we have first we can study how fast is the structural change triggered by the calcium binding site. So the calcium is represented here in the troponin C in yellow by these black dots. So the troponin in skeletal muscle binds two calcium ions, which are represented here. And here is the position of the TNC-C probe, which is the probe I was telling is in the regulatory lobe of troponin, close to the binding site of calcium. And these tell us how fast is the calcium switch in troponin C. So as you flash the fiber with UV, you have a very fast force rise, which take about 100 milliseconds to get to the plateau, to the maximum force. This is called an isometric contraction because we keep the fiber length constant. And then we measure the polarization of the probe. So again, the parameter, the angular parameter, P2. And we can see here in the inset how fast is the structural change. Because this is extremely fast. It's probably the fastest structural change that we can detect. And this is occurs in a sub-millisecond time scale. As you can see, most of the structural change happened within the first millisecond when no force has been generated, no active force has been generated following the release of calcium. So this angular change then corresponds to the structural change induced by calcium binding and by some downstream event, which basically released the tropomyosin from the blocking position. In fact, the force starts to generate 1 millisecond after the flash photolysis. So this tells you that the thin filament becomes available already after 1 millisecond after the calcium release, which is very fast. Then if we look at the downstream event, so we look at the TNC-E probe, which is the probe which is in the scaffold of troponin, you can see that also here we can detect a very fast structural change. So this fast structural change, which happens within 1 millisecond, it's not just calcium binding, it's also a structural change which is transmitted via this alpha helix to the rest of the tropomyosin. So the whole tropomyosin complex can reorient on the thin filament structure, on the thin filament axis, on a millisecond time scale. So that's a very fast structural change that no other structural approach except the fluorescent polarization can pick up, because all the structure you can see with the EM are static structures. So here we can follow very fast structural changes in the troponin complex. And then this is followed by a slower structural change, which seems to track the force generation of muscle. And in particular, I can tell you that this structural change actually tracks the myosin head binding to actin. So this tells you how fast the myosin can attach to the actin to generate the force. And then finally, we look at the regulatory structural changes that happens at the level of the myosin RLC. And so again, we use this probe in the C lobe of the RLC, which report the orientation of the myosin liver arm. So in the resting muscle, again, this probe is almost parallel to the filament axis. And then you trigger the calcium release, and nothing happened for about 3 milliseconds after the flash. So this means that the folded motor remained folded in the very first 3 milliseconds after the flash photolysis. So who is the generator of force, the force generator in this initial phase of contraction? And we think that these are motors which are not folded in the inhibited structure of myosin, but they escape this control, and they are what we call constitutively on motors. So motors that can immediately interact with actin as soon as the tropomyosin blocking position has been removed. And so this myosin motor then trigger a force-induced structural change in the myosin filament, and then you can see that slowly the myosin motor gets activated, and that's a sigmoidal time course, which is very, very different from what we've seen from the troponin. But the striking result is that while the troponin doesn't seem to limit this kinetic change in force, this structural change, especially the early phase of the structural change in the myosin motor, seems to limit the force generation, and is actually associated with the lag in force generation that we observe in the very first millisecond after the calcium release. So these results allowed us to basically define a model of filament activation in which the thin filament can get quickly activated within 1 millisecond by calcium binding, and then the myosin motor are slowly released from the folded conformation depending on the load applied on the filament. And in the first instance, the myosin motors, which are constitutively on, can trigger this stress-dependent activation by interacting with the actin filament and generate the force trigger that is necessary to generate a positive feedback loop in the activation of the myosin motor on the thick filament. So I think that concludes our journey, and I think the conclusion, the biological conclusion, is are related to the initial question of how the structural dynamics of the filaments can basically control the rate of force generation. So we can say that the thin filament activation in physiological condition in a muscle is fast, and it takes about 1 millisecond. And then the myosin motor can are activated by stress. And this is consistent with the mechanosensing mechanism that we have proposed for the activation of the thick filament. And the speed of force generation in the isometric contraction is therefore mainly limited by this stress-dependent activation of the myosin motor from the fold state. And so in our model, therefore, the filament acts as a gate. The thin filament acts as a gate because it is gating the access of myosin motor to the actin binding site in a calcium-dependent manner, whereas the thick filament mechanosensing control the number of myosin motor that can get activated, can get recruited according to the external load applied on the filament. But also this controls the speed of activation, because if you apply an external load on the muscle and on the thick filament, for example, via tightening, you can induce a faster recruitment of myosin motor, triggering a faster mechanosensing transition in the motor. So here are some, just suggest some reading that can help to understand better this phenomena and the techniques that we have presented in this seminar. And so with this, I thank you for the attention, and I welcome your question. Stop sharing, and I'll see if anyone has any question.
Luca, can you hear me?
Oh, yes, I hear you.
Hiya.
Thank you.
Thank you for the talk. Really interesting. I'll be honest, this is an area which I don't understand very much of. I'm more of a neuroscientist, so muscle is quite not my specialty.
Of course.
But I was quite curious about this, the angle changes which were leading to activation, or the on and off sort of switching.
Yeah.
I don't know whether you could bring up the presentation again, just so I can see.
Yeah. So can you see my presentation?
Yeah.
So you can tell me, see if I can go back to.
It's where you've got the acute, the obtuse angle.
Right. So
yeah. Slide 10, I believe. Or
10. Oh yes.
9.
Yeah, Stefan.
The one above.
Yeah, this one.
Yeah. I was just wondering if you could explain a bit more to me about this force generation mechanism.
Okay. Yes, sure. Actually, what I presented in the talk is mainly related to this mechanism here, which is the regulatory mechanism. I haven't said basically anything about how we use the probe to detect the force generation mechanism inside the muscle fiber. So, so I hope you understood this one.
Yep.
So
Yep,
the
that's
regulatory
fine.
mechanism off and on. So big change, angular change. So here, yeah, I think what we have to say here is that actually there are two structures for the myosin here. So one is at the beginning of the working stroke, one is at the end of the working stroke. That's achieved by crystallizing the motor in a different nucleotide state. So this is in the ADP API, so the hydrolyzed state just before the working stroke. This is the Rigor state, means with no ATP bound, which it's at the end of the working stroke. So if you overlap this structure by the catalytic domain, you see that the myosin motor has this light chain domain, so the long alpha helix domain, tilted by 70 degrees with respect to the structure in Rigor. So this means that in changing the nucleotide states, this structure has rotated by 70 degrees. That's what we call the atomic model of the working stroke. So it's the evidence of how the chemical energy of the ATP is basically converted in movement. Because if you think that this end here of the molecule is connected to the filament, you can see that this generates a sliding of about 10 nanometer. This motion here, this angular motion generates sliding of 10 nanometer. So it's a structural change that we can detect. If you place a probe here, then you can apply fast mechanical perturbation, and you can induce the tilting. So you will see the probe that will tilt from one orientation to another orientation because of this structural change. Does it make more sense to you? Or
Yes, it does. Thank you. Yeah,
yeah.
that's amazing.
Yeah, so that's the working stroke. So it's a unitary step of force generation that is produced by a single myosin motor when hydrolyzes ATP. So it's a 10 nanometer stroke, which generates a 10 nanometer sliding, relative sliding of actin by over the thick filament.
Okay.
Yeah. So
And
I have, yeah, go on.
No, I just have. I was just, if I may, one more question. I mean,
Yeah,
would you imagine that you could find this sort of mechanism in other organs or tissues or cells even? Would you expect this across many different types of cells?
Yeah, so if you think there are many, so the molecular motors is a huge family of proteins. So here we are interested in myosin 2, which is the muscle motor, muscle myosin, and also the in cardiac muscle we have myosin 2 isoform. But in every cell you have many different families of myosin, and all the myosins have they share this kind of structural change associated with ATP hydrolysis. And what is different is the energetics of the process, or how the hydrolysis is coupled to the rotation of the lever arm. Because myosin 5 has a much longer lever arm, so the same degree change in orientation lead to a much longer axial displacement. So every myosin has evolved its own mechanism, but this angular conformation, angular change in protein domains, is typical of myosin motor proteins. If you think also kinesins is another class of myosin motor that involve a rotation of domains associated with ATP hydrolysis, and the other class of motors is the dynein family, and also in those proteins there are angular structural change in domain. So I would say yes. So there are many other examples of how an angular change, angular motion of a protein coupled to the ATP hydrolysis can generate motion in cells. Yeah, so that it's a fundamental mechanism of motion.
That's amazing. Thank you. Really interesting.
We can prove that with these probes, with these bifunctional probes that you can use even at single molecule level to detect these angular changes in single cell or in single molecule assays.
Okay, thank you very much. That's
No,
great.
thank you. Thank you. Cheers.
You said at the end about how the calcium activation, the calcium signal can lead to really fast activation of the thin filament. What sort of physiological relevance do you think that has?
Yeah, so I think that's very much related to the function of fast skeletal muscle. So there are many different types of muscle. You have, first of all, differentiation in fast skeletal, slow skeletal. Cardiac muscle is another type of muscle. So what is different in all this muscle is kinetic. So how fast the structural change can be accomplished, that determines the function. So how fast the muscle can contract. So I think here we are specifically investigating the fast muscle type, which means is a muscle that is used to generate very quickly motion and force. So in this case, in these cells, then the fast activation of the thin filament is just a switch, is just a trigger for contraction. So the thin filament activation is not graded in skeletal muscle because the calcium release is massive, and so the calcium is always maximal even after a single action potential in the muscle cell. So you get a full activation probably of the thin filament, and that's also fast. So I think this has been evolved to generate a very fast response in contraction. And if you take other muscle which are not made for generating very fast motion, for example, cardiac muscle, then you have a different type of requirements, different tasks, different mechanical tasks. So in that case, calcium is not maximal. The calcium release after an action potential, it's submaximal, and so in that case, the activation kinetics of the thin filament is probably not as fast as we see in the fast skeletal muscle, and therefore is more graded to adapt basically the cardiac muscle to a different type of mechanical task. And so we see at the thick filament level, the mechanosensing is also different, has a different sensitivity, because probably in that muscle you want to tune the mechanical activation of the motor to the range of forces applied during contraction. Does that answer your question?
Yeah, that makes a lot of sense. Thank you very much. And also, out of interest, I used to work on Myosin 7, or Myosin 6, sorry, Myosin 7, Myosin 6.
Okay.
Have you ever looked at like sort of the mechanosensing mechanisms, or like any sort of like any studies there? Have you looked at other types of myosin or?
No, so far, so we have developed all these approaches to investigate what happened in muscle fiber. So we are, so our technique now is optimized for myosin 2, you would say, in the physiology, because our main interest as physiologists, biophysics, is to study those processes in the most native environment, which is the sarcomere. So you don't want to disrupt the sarcomere. You don't want to even, because most of these regulatory mechanisms can be lost if you work with single molecule, so single myosin, for example. And so we want to, so all our techniques has been optimized to perform these studies in situ in the muscle cell. But what we are trying to do now, so we are not going to single molecule level. So I think it's possible, as I said, there are fluorescent studies we're using fluorescent polarization on myosin 5, which are very interesting single molecule studies, because myosin 5 works as a single molecule. So that has been done by other labs. What we try now to do is, it's more a single molecule study, but in the sarcomere. So what we're trying to develop is an imaging method to visualize what happened to single molecule labeled with, single protein, single myosin motor labeled with our bifunctional rhodamine in the sarcomere. So we want to visualize what happened in the sarcomere, different region of the thick filament for each single motor. That's a bit more the challenge of nowadays. But we can do that. We have preliminary data on myofibrils, which are the subunit that allows us to do more super resolution studies, but still keeping the physiological organization of the filament in the sarcomere lattice. And also they allows us still to perform mechanics. So we can still have pullers and transducer to measure the force and generate stretches, and so we can see the effect, resulting effect at the level of each molecule in the sarcomere. So that's our holy grail now.
Yeah, thank you so much.
Thank you for your question.